Good morning, everyone, and welcome to today's presentation. I'm delighted to have you all here. Today, we will be discussing the qualifications and standards related to clean rooms, specifically focusing on airborne particle counts. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this session, we'll explore what makes a clean room, how airborne particles are measured, and why these standards are crucial for maintaining a sterile environment. So, let's get started and dive into this important topic together. Let's start by understanding what a clean room actually is. A clean room is a specially controlled environment where we carefully monitor and limit airborne particles to ensure contamination stays within predefined limits. This is crucial because even tiny particles can compromise product quality or safety. Moving on to the objectives of qualification, the main goal is to demonstrate compliance with regulatory standards, showing that our clean room meets all necessary requirements. We also want to verify how effective our contamination control measures are, ensuring they work as intended. Additionally, maintaining product quality and sterility assurance is central, as these are often critical in industries like pharmaceuticals and biotech. Lastly, establishing a baseline for environmental monitoring helps us track and maintain cleanliness over time. Now, the qualification process itself involves several stages. Design qualification, or DQ, ensures the design of the clean room meets all specifications. Installation qualification, IQ, confirms that the environment and equipment are installed correctly. Operational qualification, OQ, tests that the system operates as intended under normal conditions. And performance qualification, PQ, verifies that the clean room performs consistently during actual operation. A key part of the OQ and PQ stages is airborne particle count qualification. This involves measuring and classifying the cleanliness of the clean room based on the concentration of airborne particles. It's a critical component because it provides a quantitative way to assess whether the environment maintains the desired cleanliness levels, which directly impacts product quality and safety. So, understanding and controlling airborne particle counts is essential for ensuring our clean room performs at its best. When we talk about important particle sizes, we're focusing on particles that are 0.5 micrometers or larger, as they can significantly impact cleanroom performance. Larger particles, say 5 micrometers or more, are even more critical because they are easier to detect and control. These sizes are essential benchmarks for maintaining air cleanliness standards. Next, let's consider occupancy states, which refer to the different conditions a cleanroom can be in. The as-built state is when the cleanroom is first constructed. At rest means it's not in use but ready, and operational is when the cleanroom is actively being used for production or research activities. Monitoring these states helps us understand how contamination levels can change. Moving on to regulatory requirements, there are several key standards. ISO standards, specifically ISO 14644, classify air cleanliness based on particle concentration, providing a clear framework for testing and compliance. ISO 14644 focuses on ongoing monitoring to ensure continued performance. These standards are fundamental for establishing and maintaining a controlled environment. In addition to ISO, there are good manufacturing practice, or GMP, requirements. The EU GMP Annex 1, WHO GMP Guidelines, US FDA Aseptic Processing Guidance, and PIC-S guidelines all set out best practices for aseptic environments. They help ensure product safety and quality across different regulatory regions. Finally, let's touch on regulatory expectations. Before using a cleanroom, it must be qualified to confirm it meets all necessary standards. Regular requalification is also vital to verify that conditions remain consistent over time. Trend analysis and investigating any excursions or deviations are key to proactive quality control. And, of course, maintaining proper documentation and ensuring data integrity are essential to comply with regulatory expectations and to support audit readiness. All these elements work together to ensure the integrity and safety of our cleanroom environment. Let's take a closer look at the key principle here. lower ISO class number equals a cleaner environment. Essentially, regulatory standards classify clean rooms based on how many airborne particles are present per cubic meter of air. The lower the ISO number, the fewer particles there are, and the cleaner the environment. For example, in the EU GMP grading system, Grade A and Grade B correspond to ISO 5 standards, which are very clean environments suitable for critical processes. Grade C is classified as ISO 7, and Grade D as ISO 8, which are still controlled but less strict. Now, to put this into perspective with some specific numbers, the pharmaceutical standards specify maximum particle counts for each ISO class. For ISO 5, the maximum is 3,520 particles of 0.5 micrometers or larger per cubic meter. For ISO 6, it's 35,200 particles, and it increases significantly for ISO 7 and ISO 8, going up to over 3 million particles per cubic meter for ISO 8. This classification system, known as the ISO 14644, helps us understand and control the cleanliness of a cleanroom environment. It's fundamental to ensuring the quality and safety of pharmaceutical products. Remember, the lower the ISO number, the more stringent the control, and the cleaner the environment we're working to maintain. Let's walk through an example calculation to understand how we determine the maximum allowable particles for a specific ISO class. In this case, we're looking at ISO class 5, with a particle size of 0.5 micrometers. The calculation involves a couple of key variables. First, N, which is the ISO classification number, in this case 5. Second, D, the particle size in micrometers, here 0.5. Using these, we apply the formula to find Cn, the maximum permitted particle concentration in particles per cubic meter. When we do the math, it comes out to approximately 3,520 particles per cubic meter. This number is very important because it sets the acceptance criterion during the qualification process. Basically, if the particle count in the cleanroom is below this level, it passes the test for that ISO classification. Now let's talk about the formula itself. It's called the particle count classification formula. It calculates the maximum allowable concentration based on the ISO class number and particle size. The formula is Cn equals 10 raised to the nth power, multiplied by the ratio of 0.1 over d, raised to the power of 0.8. This formula is fundamental in ISO 14640-41 standards for cleanroom classification. It helps us ensure that our environments meet strict cleanliness requirements by quantifying the maximum allowable particles for different classes and particle sizes. On this slide, we're looking at the typical parameters we record during particle size analysis. First, the particle size channel is crucial because it categorizes particles based on their size, which helps us understand the distribution within a sample. Then, we have the sample volume, which is simply the amount of air or fluid we analyze, ensuring consistency across measurements. The location ID and date or time are essential for traceability and for correlating results with specific environmental conditions or sampling periods. Operator identification is also recorded to maintain accountability and quality control. Moving on to additional environmental parameters, these factors, such as differential pressure, temperature, relative humidity, and air velocity, can all influence particle measurements. Monitoring these helps us interpret results accurately and understand the environmental context during sampling. Now, let's touch on the instrumentation involved. The main equipment includes the laser particle counter, or LPC, which is the core device for counting and sizing particles. We also use an isokinetic sampling probe to ensure that particles are collected at a consistent velocity, matching the airflow. Tubing and flow controllers are vital for directing and regulating the sample flow, and a calibration standard ensures the equipment remains accurate over time. Speaking of calibration, it's very important that our instruments are calibrated according to ISO 21501 standards. This guarantees traceability and consistency. A calibration certificate proves that the device has been tested and verified. Lastly, the flow rate typically needs to be around 28.3 liters per minute, which is a standard flow rate to ensure reliable and comparable results across different tests and setups. All these parameters and equipment considerations are fundamental to maintaining the accuracy, reliability, and traceability of our particle measurement process. Let's walk through an example to see how we determine sampling locations in a cleanroom. Suppose the cleanroom area is 64 square meters. To figure out the minimum number of sampling points needed, we use the simple calculation. Take the square root of the area. So, in this case, the square root of 64 is 8. That means we need at least 8 sampling locations to properly cover the space. When considering where to place these sampling locations, we focus on key areas such as critical processing zones, operator activity zones, air return points, and worst-case contamination spots. The idea is that sampling locations should represent the entire cleanroom environment, capturing any potential contamination sources. Now, how do we determine the exact number of locations? We use the ISO 14644 formula, which is quite straightforward. The minimum number of sampling locations equals the square root of the cleanroom area in square meters. And remember, always round up to the next whole number if you get a decimal. This ensures we don't underestimate and miss any potential contamination risks. So, in summary, by calculating and strategically placing these sampling locations, we ensure thorough monitoring of the entire cleanroom environment, maintaining the quality and safety standards we aim for. Let's look at this example. The ISO 5 limit at particles greater than or equal to 5 micrometers is 29 particles per cubic meter. To determine the sampling volume, we perform this calculation. V's equals 20 divided by 29, multiplied by 1,000. That gives us approximately 689.65 liters. Now, interpreting this practically, if we're sampling at a flow rate of 28.3 liters per minute, the sampling time would be roughly 24 minutes per location. This duration helps us achieve a reliable statistical representation of the particle concentration. Moving on to the sample volume calculation, the minimum single sample volume is determined by this formula. V's equals 20 divided by Cn, m, multiplied by 1,000. Here, V's is the minimum sample volume in liters, and Cnm is the class limit for the largest particle size we're considering. Using this approach ensures that our sampling process maintains statistical confidence, which is crucial for accurate qualification and compliance. This way, we can be confident that our results truly reflect the particle levels in the environment. Now, let's move on to some important precautions to ensure accurate sampling and reliable results. First, always avoid probe obstruction, because any blockage can skew the particle count readings. Next, use isokinetic sampling, which means maintaining the same velocity of air entering the probe as in the environment to get representative samples. It's also crucial to prevent tubing bends, as sharp bends can create turbulence and affect the flow. And finally, maintain strict aseptic practices throughout the process to avoid contamination that could compromise the test. Moving on, we have a table defining different states of equipment and environment during testing. The as-built state refers to when the equipment is installed but not operated. The at-rest state is when the equipment is running but no operators are present. And the operational state is during normal production activity, when everything is functioning as it normally would. Now, let's look at the airborne particle count test procedure. It starts with verifying the calibration status of the instrument to ensure accuracy. Then, we record the environmental conditions, like temperature and humidity, which can influence particle counts. Next, it's important to confirm that the HVAC system has stabilized, so airflow and environmental conditions are consistent. We also define the occupancy condition, whether the area is occupied or unoccupied during testing. Following that, we perform the particle count sampling at each designated location, making sure to record the counts accurately. We then compare these counts against the acceptance criteria established for your specific environment. Lastly, we generate a qualification report summarizing all the findings. Regarding testing conditions, it's essential to follow a consistent and controlled approach, ensuring the environment is stable and representative of normal operation. These steps help us validate that the environment meets the required cleanliness standards, supporting overall quality and compliance. In this slide, we're looking at our recent observation, which confirms that all locations meet the ISO 7 acceptance criteria. This is an important point because it shows our cleanroom environment is within the required standards for particle counts. Moving on to the interpretation, the data indicates that our HVAC system is performing effectively, maintaining proper airflow and filtration. The air filtration itself is adequate, ensuring contaminants are kept in check. Overall, the cleanroom is acceptable for its intended operation, which is critical for maintaining product quality and compliance. Let's take a quick look at the table. For each location, from L1 to L5, you can see the particle counts for particles of 0.5 micrometers or larger. The counts range from 120,000 up to 310,000 particles per cubic meter. Importantly, all these values are well below the acceptance limit of 352,000 particles per cubic meter, so each location has passed the qualification criteria. This example of qualification data demonstrates that even at rest, the airborne particle counts stay within acceptable limits, reinforcing the integrity of our cleanroom environment. Overall, these results give us confidence that our systems are functioning as intended and that we are maintaining the strict standards required for our operations. Now let's talk about the qualification decision. Essentially, if all locations are within the specified limits, the result is a pass. But if any location exceeds those limits, then it's a fail. Moving on to the common causes of failure, there are several factors we should be aware of. These include HEPA filter leakage, which can compromise air purity. Improper airflow balancing can create uneven conditions, and excessive personnel movement can disturb the controlled environment. Inadequate cleaning is another critical factor, as contaminants can accumulate. HVAC malfunctions can also lead to environmental instability. Let's look at the table, which shows the regulatory acceptance limits for particle counts under different grades. For Grade A environments, at rest or in operation, the limit is 3,520 particles of 0.5 micrometers or larger. Grade B has the same at-rest limit, but in operation, it's allowed up to 352,000 particles. Grade C allows for higher limits, 352,000 at rest and up to 3,520,000 in operation. Grade D is more lenient at rest with a limit of 3,520,000, but in operation, it's not defined, meaning it generally isn't suitable for critical environments. These limits are aligned with EU GMP Annex 1 standards, which set the regulatory acceptance thresholds. Understanding these values helps us ensure that our environments meet the necessary cleanliness standards for compliance and safety. When it comes to regulatory agencies, they expect us to conduct investigations that are thoroughly justified by scientific evidence. This means we need to provide clear documentation at every step. First, if there's a deviation, we must prepare a deviation report that details what went wrong. Then, we perform a root cause analysis to understand the underlying reason for the deviation. Once we identify the cause, we implement corrective and preventive actions, or CAPAs, to address the issue and prevent it from happening again. Examples of typical CAPAs include replacing a HEPA filter, rebalance airflow, retrain personnel, revise cleaning SOPs, or increase monitoring frequency. These steps are crucial to maintaining a controlled environment and ensuring quality. Handling qualification failures involves a structured investigation process. We start by reviewing raw particle count data to see if there are anomalies. Then, verify that our instruments are properly calibrated. Assessing HVAC performance is also key, checking airflow, pressure, and filtration efficiency. Inspecting the HEPA filter integrity is essential to confirm it's functioning correctly. Evaluating personnel practices helps identify if human error contributed. Lastly, reviewing cleaning records ensures that cleaning procedures are being followed consistently. All of these steps and documentation efforts are vital because they demonstrate our commitment to compliance and continuous improvement, which is the core message of this presentation. Now, let's take a closer look at modern monitoring systems. These are essential for maintaining a safe and controlled environment. First, we have real-time particle monitoring, which allows us to continuously track airborne particles as they happen. This immediate data helps us respond quickly to any issues. Next, we rely on data logging software. This technology records all our monitoring data over time, giving us a valuable history to analyze trends and make informed decisions. Remote alarm systems are also crucial. They alert us instantly if any parameter exceeds safe limits, even if we're not physically on site. Additionally, automated trend reports compile our data into clear, actionable insights, making it easier to spot patterns or emerging problems. Now, moving to the table, we see different levels and their purposes. The alert limit serves as an early warning system. It's our first sign that something might be amiss. The action limit, on the other hand, prompts immediate investigation when exceeded, helping us prevent contamination or process failure. Our routine monitoring covers key parameters like non-viable particles, viable airborne counts, differential pressure, temperature, and humidity. These measurements are fundamental to ensuring a controlled environment. When it comes to trend analysis, the goals are quite strategic. We want to detect gradual deterioration early, predict potential HVAC issues before they escalate, and maintain a consistent state of control. All of this supports our broader contamination control strategy. Finally, understanding alert and action limits is critical. They define our thresholds when alarms trigger, whether for early warnings or immediate action, helping us stay proactive rather than reactive in maintaining environment quality. Let's start by looking at the typical requalification frequency. Generally, we aim to requalify equipment every 6 to 12 months to ensure ongoing performance and compliance. Of course, after any major maintenance or modifications, requalification is also essential to verify that everything remains within specifications. Moving on to documentation and compliance expectations, this is where things get really critical. We need to adhere to ALCOA Plus principles, which emphasize data being attributable, legible, contemporaneous, original, accurate, complete, consistent, and enduring. An electronic audit trail is vital for tracking all data changes, and controlled worksheets help maintain data integrity. Reviewer approval is also required to ensure oversight and accountability at every step. Now, let's talk about the required documents for a proper qualification process. These include the qualification protocol, which lays out our plan, the approved test procedures, raw data sheets capturing the actual results, calibration certificates validating our equipment, the final qualification report summarizing everything, and deviation and CAPA records to document and address any issues that arise. When it comes to regulatory inspection focus, inspectors are particularly interested in data traceability, being able to follow every piece of data back to its source. They look for a solid scientific rationale behind our methods and trending evidence that supports consistent performance over time. Requalification frequency is also scrutinized, as is our contamination control strategy, or CCS, which is crucial for maintaining product integrity. In summary, maintaining a rigorous requalification schedule, thorough documentation, and a strong compliance mindset are all key to passing inspections and ensuring quality in our operations. In conclusion, understanding and implementing airborne particle count qualification is absolutely essential for maintaining cleanroom compliance. This process ensures that the environment consistently meets the strict standards set by regulatory bodies. As we've discussed, frameworks like ISO 14644 and EU GMP Annex 1 provide the necessary guidance and requirements to follow. It's also crucial to develop a proper sampling strategy and perform accurate calculations. as these are key to obtaining reliable data. Qualification not only demonstrates that the HVAC systems and contamination control measures are effective, but also builds confidence that the cleanroom remains safe and compliant over time. Continuous monitoring plays a vital role here, ensuring ongoing compliance and early detection of any issues. Overall, effective cleanroom qualification minimizes contamination risks, supports regulatory adherence, and ultimately protects product quality and patient safety. The references we've referred to, including ISO 14644-1 2015, ISO 14644-2, EU GMP Annex 1, FDA guidance, and WHO GMP guidelines, serve as essential sources for aligning technical practices with international standards. Remember, maintaining this rigorous approach is fundamental to the integrity and success of our cleanroom operations. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional guidance you might need. Remember, the goal is to ensure you feel confident and well-equipped moving forward. Thank you so much for your time and attention today. Your engagement is truly appreciated.